Good morning and welcome to webinar 4 in the Lighter Touch Soilborne Disease series. My name is Jeff Smith and I'm the technical lead for the agroecology projects at Lighter Touch. Today's focus is on the future innovations in diagnostics and plant breeding, and we will round out the series with a look at the Lighter Touch Soilborne Disease project with the New Zealand Buttercup Squash Council. We have 4 speakers in today's program and we will pause for questions after these first 2 presentations. Please ask questions during the presentations via the Q&A function, and perhaps preface your question with the presenter's name so that we can direct it to the right person. Everyone who is registered to attend will be emailed the recordings when available online. So without further ado, I'd like to introduce our first speaker, who is Mark Braithwaite. Mark is a diagnostic plant pathologist trained in both classical, morphological, and modern molecular identification methods. Mark has 40 years experience identifying fungi and bacteria for growers, industry representatives, and quarantine biosecurity purposes. In addition, Mark has experience in designing and conducting research trials, surveying crops for plant diseases, fungicide resistance screening, and providing advice to plant health problems. Welcome, Mark.
Oh, thank you, Jeff, and welcome everybody. So I'm really going to be talking about diagnosing soilborne disease issues, how we are currently doing that, and what is going to be like for the future. So next slide. So just by way of introduction, Plant Diagnostics is a family-owned business in Christchurch. Our focus is on conventional diagnoses, using things like agar plating, identification by morphology, but we do delve into DNA sequencing where necessary, and also some PCR testing, which we'll talk about. So I believe this provides a holistic approach to plant disease issues using our plant pathology expertise, along with, as I said, the conventional and molecular methods. So the scope of this talk really is looking at fungi and oomycetes, oomycetes being Phytophthora and related genuses. But a little bit on viruses, but it won't be including things like nematology or other causes of soilborne disease. Next. So the way I'm going to approach this is that we're going to look at how we diagnose a disease problem. And I'm using avocados as an example here. And then we'll just go through the methods that we use to diagnose soilborne disease issues, and then what's happening next. So slide. So the traditional approach to disease diagnostics is, one, we're presented with a problem. This might be sick plants or a whole paddock, it might be one plant. So what we need to do is triage the symptoms that we've got before us. Is it the leaves? Is it the roots? Or, what symptoms can we actually see? We then culture from these areas to agar, if that's possible. And then if we're getting fungi out on agar, we then can look at the morphology of the fungus. This is like the microscope type work. And if we can't take it further from there, we will look at sequencing and potentially phylogenetics, which is just grouping fungi into their families, which I'll show you. And last but not least, really, is pathogenicity testing. I mean, if we've isolated these organisms, do they actually cause disease? Because that's the big question. Next. So just looking at a case study we did with avocados, the industry was confronted with an issue. Newly planted plants were not doing well. They were dying early or struggling along. So it's quite a good example to show, what the process is to determine the problem, and then it gives you options to how to solve it. So next slide, please. So the problem was these plants, when we dig them up, there's a lot of root rot going on. You can see on the right there, there's in the centre, there's lots of root blackening and browning lesions and a lot of loss of root architecture. And some of the plants also, if you look at the slide on the picture on the left, there's a lot of staining at the base of the plant. And I've just thought I'd talk about sampling of plants with disease in the base, like cankers. And the way to do that is that you need to cut into it and look for the junction between the healthy and the diseased tissue, because that's where we're most likely to isolate the fungi or whatever it is associated with the issue. So you can do this by getting out your chainsaw and taking a wedge out. You can drill into it and take shavings, or if it's small enough like these plants, you can dig up the whole thing and send it in for diagnosis. So there's lots of options. Next slide, please. So what we did is we isolated from the base of the trunk the roots, and we got a whole lot of different organisms. The most dominant being Aotearoa dactylonectria, which used to be called cylindrocarpal. Everything changes. There was also a reasonable amount of fusarium and pythium, and then a whole range of smaller or lesser ones present, such as Phytophthora and other organisms. So we sort of focused really on the Aotearoa because that was the dominant organism that came out. So next slide, please. So what we wanted to do is how do we identify what's going on with these plants? So we sequenced a whole lot of isolates, and John will talk to you probably about what a sequence is. And basically from that sequence, which is a DNA sequence, we can build what we call a phylogenetic or family tree. So how are these species interrelated? And we can get a much better fix on the identification of the organism. So with these particular ones, 90% of them were Aotearoa liriodendri, and there were a smattering of other ones. That tree there, the one on the part to the left, actually just sits on top of the part to the right, so it's a continuous tree. It just doesn't fit on a page, that's it. So this was the start of what we were trying to do to identify what was going on, and Aotearoa liriodendri was very consistently coming out. So next slide, please. So just to look at what do these organisms look like on an agar plate. So that's along the top there, and you can see the associated spore types down the bottom. So morphologically, there's not a lot of difference, particularly between liriodendri and pinicola. If you looked at them on an agar plate, it's very hard to tell the difference. So that's where the DNA sequencing comes in. Next, please. So what we did then was to set up a pot trial, and using these isolates, a cohort of them, we inoculated young avocado seedlings, let them grow, and allowed the disease, if it was going to develop. And this was over about 6 weeks. So as you can see, if we look at the plant on the left there, it was healthy. It wasn't, no fungi was put into there, and so the roots look relatively healthy. There's good top growth. Some plants were showing sort of mild to moderate symptoms, so there was a height reduction and obviously a bit of loss of root architecture and also the root browning. But the plant on the right was a massive loss of root structure and virtually no roots left. In fact, probably that plant's only surviving what's left in the seed, and obviously a big reduction in top symptoms. So obviously some of these organisms were definitely causing disease. Next slide, please. And as you can see, this is just a bar graph showing the disease score, how much disease did it have, and the different isolates that we used across the bottom. And isolate 311, as you can see, was causing significantly more disease than any of the other isolates. And in fact, some isolates, the ones out to the right there, were not causing any disease at all. And the negative control had a slight bit of disease in it, probably because, it's hard to get plants that don't have any disease. But certainly, I think this definitely proved that particularly that isolate 311 was causing quite significant disease. I haven't presented the data here, but also it reduced significantly the root dry weight and also as you could see, for the plant height. Next, please. So to actually fulfill Koch's postulates, which is something that that's important in plant pathology, you've got to re-isolate the organism from the one that you've inoculated with the disease. So we did this, and out came Aotearoa liriodendri, and we also tested it or resequenced it, and it was the correct species. So the key findings here is that the pathogenicity wasn't consistent across isolates, and that was sort of an interesting find, really. One isolate was particularly bad, others caused some disease, but not as much as that particular isolate. So obviously it seems to be strain dependent and not necessarily genus dependent. So what's required in this instance is that we need to do more genetic work to identify why is that isolate pathogenic, and can we develop a test that would detect it, like a qPCR test in the soil? Because if we've targeted the actual pathogen, the one that's causing disease, then that's the best that we can do to, for instance, develop a pre-plant test. And as I said, some of them actually didn't cause any disease at all. So they're just there and probably beneficial in the environment. Next, please. So there is some relevance to the current soil-borne issues on squash with what we've done with the avocado. We could take a similar approach and identify everything that's associated with the roots, and then do pathogenicity studies and find out which ones are actually causing disease, because that's what's important. And from that, there's the potential to develop a more specific PCR test that can detect these pathogenic strains. So it might be the genes that cause the organism to be pathogenic on plants would be the better target than the organism itself. Next slide, please. So part two, going back to methods. So one method that we use quite often actually is soil baiting. And it's really good for organisms like mycetes, Phytophthora, Pythium, Phytopythium, because they produce a zoospore, which you've probably heard about, that swims in water and infects plant roots. And to do this, we bait with things like cedar needles. We can cut leaf discs out, like rhododendron leaves. Or we can use whole plants like strawberry seedlings, just to use as a bait to attract these zoospores. So there's an example of a Phytophthora growing on agar and the type of sporangia that it produces. Next slide, please. So I've got a wee treat here for you, so I need to click it again. This is showing you the release of zoospores from a sporangium. So if you just watch that sporangium in the centre of the field, you can see them now pouring out. So the spores have been released and they're on their way looking for roots to infect already. As you can see, they move pretty quick. Next slide, please. Right. So going back to methods again, so I just wanted to briefly also again look at the traditional methods that we use. So culturing, it's a bit slower than other methods, like your DNA methods, but it does provide cultures of fungi and bacteria that you can then further analyse, like we did with the avocados, either by sequencing or next generation sequencing. And the other advantage is that it does detect live organisms, so it's not detecting anything that's dead or anything which some of these other methods do. So it's providing real life scenario situations. And it provides a very broad screen of the cultural organisms that we've got. So, you can, just because you're plating on an agar, you can detect a lot of different things at the same time. So I've just got an example here. Recently we received black currant that was in a paddock that was, a lot of plants were not looking too good actually. So there was when we got it, there was a root rot in the base of the plant, and as you can see that in that photo there out to the right was the stem of the plant, which was virtually completely dead. And there's a nice junction between the healthy and diseased. So next slide, please. So what do we find? Well, what didn't we find was the question, actually. So from the top, we got a fungus called Aotearoa lata, which is a serious pathogen on grapes and also on apricots and other stone fruits and other woody plants, actually. So it is quite a serious pathogen. So it was associated with that top-down dieback. And the roots, we got Pythium, Fusarium, and Aotearoa again. So there's a whole lot of things going on in the roots. And then in the soil, we actually baited Phytophthora out of the soil. So sometimes it's hard to get Phytophthora from the actual roots because it easily gets overrun with all those other organisms that we found there. And also the soil was heavily infested with nematodes. So what's going on here? Well, actually, delving into this, it's very difficult to know what the actual primary cause is going on, but obviously there's a host, and it's a real host of problems, and it's a real disease complex issue, not just one thing. So it just sort of shows, what you can detect. Next, please. So looking at other testing in the soil, it's not something that we offer, but we are certainly looking into it, and it's probably something that we intend in the future. And that is molecular testing or DNA testing for plant pathogens in the soil. So I just sort of briefly talk on soil sampling. I'm not sure if John's covering it, he might be, but it's just brief. So if we are looking at soil sampling, what we do is divide the paddock into a grid, maybe 30 grids, and then you would sample with a core from each grid, so you've got a good representative sample of the whole paddock, not just the corner where you drive in. So it might get a better result out of it. The thing about qPCR or PCR of soil is it's sensitive and it's highly specific. It does require DNA extraction, so maybe it's more confined to the laboratory. It's useful for a specific target. I've just got here Plasmodium, which is club root, so that's going to be a pathogen. So if you can detect that particular organism in the soil, you probably know you've got issues. So something like that, it's quite good for. Also, we'll detect uncultural organisms. And the only issues really is that some targets may or may not correlate with plant disease, and that's what we sort of touched on with the avocados. There's a lot of other things in the soil that you just don't know what they're actually doing. It's like non-pathogenic strains like Fusarium roxysporum, for instance, and Aotearoa, there are some there which don't actually cause disease. So to develop these tests, you need a lot of research into relating it to the disease impact and the amount of soil levels, the amount of DNA levels in the soil. So identification of a fungus does not mean that it's a pathogen, and it also doesn't mean that it's viable. So there's lots of things to consider. Next, please. So looking at endpoint PCR or qPCR for isolating of soil-borne pathogens from plant tissue, and I've just got an example here, which is powdery scab versus common scab. Powdery scab is normally easy to identify down the microscope, but there's always situations where it might be a very early infection and you can't see any spores, so you're never quite sure whether it is or it isn't. So there's that issue. So therefore, the PCR comes into its own in that situation. So this test that we've got identifies powdery scab in conjunction with common scab, so it's a two-prong approach. So common scab, it's a difficult one to identify sometimes because it's not easy you can't really see it down the microscope because it's so tiny. It's more of a bacteria than the fungus. But this test actually only detects pathogenic strains. So if you get it, then you know that it is actually causing that symptom. Recently, we had what I thought was a common scab sample, and it came up negative for common scab, which was quite surprising. So we repeated, and it was still negative. So what was going on here? So we isolated, and it turned out that there was Streptomyces in it, but it was obviously a non-pathogenic strain, and we couldn't really get to the bottom of it other than maybe suggesting it was a physiological condition, and there were just strains present that were there but not causing disease. Next. So LAMP, this is another test. So it's sort of, for me, it's an alternative to PCR. Its advantages are that it's just a crude DNA extraction and it can be done in the field, so it could be fast and it does have a place, but it's not something that we currently use. I guess the disadvantage, it's a single target and it may or may not be quantitative, but it's something that's out there that you may have heard of. Next, please. So ELISA, which has been around for many, many years and is a very laboratory stable test that we currently do. It's not so much related to plant soilborne diseases, but it could be if, for instance, there's a soilborne virus that might be in the plant. It could be useful for detecting that. So it's high throughput. It's less sensitive than PCR, but it's still good enough and for most situations. And as I said, it's often used in virus detection. Next, please. So where to from here? I just thought I'd very briefly dwell on next generation sequencing because it's something that's probably going to make a move in the future. So it's rapidly evolving. Potential for sequencing a large range of organisms in a sample all at once. So it's a big advantage there. There's two main strategies, metagenomics and metabarcoding. And currently they're sort of useful for biosecurity screening of samples, environmental monitoring, community profiling. As far as I'm aware, it's not widely used for specific plant diagnostics currently, but I'm sure it will be developed in the future. Species identification may confirm pathogenicity, which is a risk with all molecular work unless you've got the right target, gene target. Next, please. So metagenomics, just delving a wee bit deeper, that's sort of sequences everything that's there, and actually including the host material. So yeah, there's a disadvantage in that, which I'll talk about. So pathogen discovery, so detect emerging or something that's unexpected. For instance, if you do a sequencing program, you might detect a new virus, for instance. It's obviously a whole genome reconstruction, so its sensitivity is high. It's often used in microbiome analysis, like how communities interact, and detecting mixed infections in a plant, for instance, could be useful. Funny thing is that there's a high host background, so that can sometimes overwhelm pathogens which might be there at low numbers. And it has a high bioinformatics cost, and that's like analyses of the data. There's a high cost involved in that, very time consuming. And, interpretation might be complex. Can have sensitivity issues. Pathogens in low numbers, as I said, might be missed. Next, please. So metabarcoding, that is being used for community profiling. So instead of doing a whole genome sequencing of everything that's there, so the target sequencing using a PCR, usually genes like ITS. So it's good for community profiling and microbiome assays, maybe environmental DNA surveillance, and it's a lot more cost effective than metagenomics. Limitations, that can have a primer bias, so some organisms might be better at others in amplification. There's potential resolution limits. I mean, can you detect between very close related species? That will probably improve. And it obviously needs a reference database. So yeah, it's just a very brief introduction. I mean, John might have more to say on that, I'm not sure, but yeah, so just in conclusion, there's a large variety of traditional and molecular methods available currently for disease diagnostics. Each method has its own merit, and the techniques and platforms, likely to become more mainstream for things like next generation sequencing in the future. But we must not lose sight of what a pathogen actually is. Simply a good identification may not quite be enough. Thank you.
Great. Thanks, Mark.
Okay.
Really, really impressive how much we do know, but
That
challenging
is. Yeah,
on what we
true.
don't know.
Well,
Yeah,
you've always got to be wary what you don't know, actually, which is a problem.
yeah, great. Thanks, Mark. We're going to move straight into our second speaker, who's John Mackay. John's coming to us from Northern Europe tonight. Tonight, your time, John. So thank you for joining late in the day. John has been working in the PCR area for over 30 years and has been developing and optimizing real-time PCR and molecular diagnostic assays for most of them. Over the years, John has developed diagnostic qPCR assays and DNA, RNA extraction methods for a range of primary industry applications. Examples of this work include the PSA-3 for kiwi fruit and more recently a test for the newly described facial eczema fungus work. He's also been working on a novel biopesticide product for the Light of Touch program and a new bee disease protocol. So welcome, John.
Fantastic. Thank you very much, Jeff, and kia ora, everyone. So, yes, in Northern in Europe at the moment, actually to talk about our bee disease we've been working on, which I'll actually touch a bit on later that I'm presenting tomorrow morning, European time. But onto soil-borne disease testing, I'm just going to talk a bit about the qPCR assay that Mark touched on really well to introduce the field. So next slide, please. So people ask us in Nature, Mark introduced Plant Diagnostics, a great company that can do many things that we can't do to Nature. But Nature is DNA in a range of nature. And so people used to ask us, what do you work on? And it's not that we got sick of that question. We always welcome that question. But we actually got a local artist to put together a piece of art. And so we'd sort of ask people to look at something in the picture they liked. And if there's something in there they liked, we'd say we'd work on it. See, as Jeff mentioned in the introduction, we started with PSA, but we work on beer, wine, truffles, Manuka honey, bee diseases, and various other things, including facial eczema, and more recently, some work on soil-borne disease fusarium. Next slide. So I'm just going to go over a bit of how PCR works, because especially in the field, you get numbers in a range of primary industries now. So a range of numbers that are generated by these methods. But how do you go from a sample of something to this number of DNA copies? So I'm going to explain a bit about PCR. And so something that's been known for a number of years, this is the early 70s, that if we had a DNA sequence on the bottom there, GAC, TATT, etc., we could design a synthetic piece of DNA called a primer that would bind based on the GCAT binding pattern. So we could use the DNA sequence on the bottom to design that synthetic primer. Next slide. And so in the early 1980s, Carrie Mullis realized that, next slide, if you did this at the same time and added 2 primers, that you could actually copy both strands at the same time as a sort of check for each strand. And no one had really thought of this idea. And the famous story is that Carrie Mullis was on his way to his weekend cabin in California. And he was thinking about work. And so he was thinking about this process. And as he thought of this idea, next slide. Next slide. He was suddenly thinking, that's not going to work. It's going to cause a problem. Those 2 primers are going to cross over each other. And it was the classic eureka moment while he's driving. He suddenly pulls over to the side of the road and realizes that he's actually copied the DNA, which doesn't really sound that exciting, I'll grant you. Next slide. And so this was in 1983. So, a few weeks ago, we marked International PCR Day, as we've done for the past few years, because that's just the tragic way we roll. Next slide. And so what PCR is doing here is, you can see the famous double helix. I don't know if my pointer is going to work, but you can see the famous double helix. If we unwind that, we'll get this ladder shape at the top there, marked target sequence. So if we go to the next slide. So PCR mechanically is quite simple, especially when Mark and I have machines that will automate this for us, where it heats the DNA up to 95 degrees and it splits apart. Next slide. The two primers that we've designed, and that's something we specialize in at dnature, is designing these qPCR assays. We cool it down and the two primers will bind if they find that target sequence to bind to. Next slide. The same way that DNA is copied in our cells when our cells reproduce, the enzyme will copy from the ends of the primers, and next slide, result in a doubling of the DNA. So when people heard this idea, they thought, big deal, you've just made another copy of DNA. But what Kerry Mullis realized on that sort of fateful drive, next slide, was that if you did it again, it would double to 4 copies. If you did it again, next slide, it would go to 8 copies. And again, next slide. If you did it 30 times, you wouldn't get 60 copies, you'd get 2 to the power of 30, which is a billion-fold increase. You've now got a lot of that amplified DNA sequence that you can then go on detect. And next slide. If you've seen something like CSI, you've seen it where they hold the gel up and say, look, we've found the DNA of the criminal, and they're pointing to bands on a gel. And this is still run in many research laboratories. But what we do now more commonly, next slide, is we use a method developed by this man, Russell Gucci. Next slide. And this is actually Russell at the Alan Wilson Centre in Palmerston North, where Alan Wilson was a famous New Zealand scientist who worked in California and was Russell's postdoc supervisor. So next slide. Russell devised a way to follow the PCR fluorescently. And what it meant, next slide, was that as the PCR was copying the DNA, it would generate fluorescence. So this is in the very early 1990s, using this ancient computer to monitor, to see the fluorescence accumulating. Next slide. And so instead of the bands on a gel at the top left there, we now get these fluorescent curves. And the beauty of these fluorescent curves is, one, we don't need to open the tubes to run a gel. So even though this is a more advanced form of PCR, known as qPCR or real-time PCR, because we can monitor it in real time, because it's much less labor, it actually works out to a much faster and thus cheaper way to detect DNA sequences. So next slide. So what we're interested in is where the DNA start to appear from that flat line. And the earlier we see so each line there is representing a single sample. And the earlier we see that fluorescence kick up there into the exponential phase is directly proportional to the amount of target that's present. So for instance, beekeepers are very familiar with this term CQ because all Manuka honey that gets exported in the pot overseas, all Manuka honey from New Zealand has to get a Manuka DNA test. And all the honey has to have a certain CQ, less than 36. So there are some primary industry applications that are becoming very familiar with qPCR. Next slide. So what we will do is we will turn that CQ using some fairly simple algorithms. We'll turn that CQ into an estimation of DNA copies. And I'll show you what that looks like shortly. But what we're actually doing is we're first having to extract DNA from the soil. And you might think of a soil sample as looking something like this, which has come, out of a kora. But with the next slide, this is more what it looks like. And one of the limitations really with soil testing is that tube is life size. That's actual size. And that's the amount of soil that we can typically process, or is the amount of soil that is typically processed. We use about twice that amount in a modification we've made to a method, but typically that is the most common amount of soil. So while you may have a paddock, a field that has different topography, and I'll talk about the limitations a bit later, while you may have a paddock that has different features, different topography, different altitudes, etc., the issue is that typically we can only really process a small amount of soil in a single DNA extraction. So next slide. Soil is full of things like humic acids and things that PCR really doesn't like very much. And so soil is an amazingly inhibitory substance. The words there aren't important. You don't need to strain to read those. It's just to show that there are a number of steps typically that go through extracting DNA from soil. It is one of the more difficult samples to do in a robust and high throughput fashion. But at the end, we basically end up with about 0.1 of a mil of water. And no matter what we're extracting DNA from, the DNA always ends up the same. We don't know whether our target sequence is present, but we have to extract DNA according to protocols that we have. And so Mark will have certain protocols for different targets in his laboratory, as we do in our laboratory at dnature in Gisborne. Next slide. So here's some work here, and this is some numbers generated, and Dereck will talk more about some of the work around squash later. But you can see here that we've got some columns there of some different fusarium species, and this ties in really well to what Mark was talking about a little earlier. So you can see here that we've got total fusarium, estimated cells per gram of soil, Fusarium avineum, Fusarium oxysporum, and Fusarium solani. And the thing is, we get very, very precise numbers there. That's a very reproducible number, PCR-wise. But the fact is, it still raises the question, what do they mean? What we often tend to take from these numbers, especially from soil, is that, for instance, between samples 3 and 4 there, one's 50,000, one's 222,000. Excuse me. Rather than looking at the absolute number, we might say there's 4-fold more total fusarium in the soil for this sample in sample 4 than in sample 3. So it does come back to, what do these numbers mean? Next slide. And so we have things like the specific target species. So we looked earlier at Fusarium oxysporum, but a bit like Mark was talking about with the cylindrocarpa, the Elenectria, he had different isolates that had different pathogenicity. Just by looking at Fusarium oxysporum is not enough to show what might be affecting the crop of interest. As Mark mentioned, we need to target that particular pathogenic species. And so while a lot of people look at DNA as the answer, DNA testing is really just another tool in the toolbox. And again, really highlighted nicely by Mark as alongside things like isolating the pathogens of interest. Next slide. So another limitation that I've already mentioned is the sample size. Actually, physically, how much soil can we put through a single DNA extraction? Where you've got things like different topography, if you've got small mounds or depressions in soil. So, for instance, it was interesting, Mark touched on, the avocado root rot. I remember working on that fungus in grapevines, and, it likes damp soil. So where you see depressions in soil, the levels of fungus in depressions might be different to, areas that are more elevated or rounded where water drains better. So again, touching on Mark's point of trying to get a representative sample, that's true, but we are limited as to how much soil we can actually put through a single extraction. Next movement. And so that comes to representation across the paddock, taking that representative sample that Mark alluded to, using grids or W patterns across a field. Next, please. So there was, I seem to have lost a slide where I was going to touch on the bee disease we work on. We look for a specific bacteria there, and having done the research that is required, we know very precisely as to what levels cause a clinical sign in beehives for this particular disease. So that's work that still needs to be done for some of these soil-borne diseases, isolating the pathogen that Mark alluded to, but then, our specialty developing diagnostic qPCR assays for laboratories to use to rapidly monitor for these pathogens. So thanks very much for listening.
Great, thanks, John. Really good roundup of what you do and how we could use it. Acknowledging those limitations that you've described. So I've got one question here for you guys, actually directed to Mark. How did you calculate the percentage of presence of various genera as you have shown on your slide from your microscopic examination? The percentage of presence of various genera.
Well, it was just really the number of isolates that we detected, actually. So it was just straight numbers out of 100.
Okay, cool. That's the only question at the moment. But if you guys stay on for the call, questions will continue to come through. So we'll come back to you. Thank you very much, guys. It's a really good roundup of diagnostics and how we're using that in our projects, and we particularly appreciate the work you've done for our Lighter Touch projects over the last years. Our next presentation is from Hugh Richardson of Enza, Zaden. Hugh's actually recorded for us yesterday due to a travel clash with today's timings. But please, again, ask any questions via the Q&A to Hugh, and we will endeavour to get those answered in writing and post those when the webinar is published. So just to introduce Hugh Richardson is a plant breeder and product developer with nearly 10 seasons of hands-on experience in Buttercup squash breeding at Enza, Zaden. Based in Pukekohe, Hugh's experience bridges practical field work, molecular breeding, and digital phenotyping. He is passionate about connecting breeding innovation with grower needs to deliver practical, high-performing variety solutions. He was a strong advocate for the role of plant breeding in addressing challenges such as soilborne disease and buttercup squash, emphasising the importance of integrated cross-functional approaches to sustainable crop improvement. So interested to hear Hugh's presentation.
I'd like to touch. It's great to be part of this webinar series. So by way of introduction, my name is Hugh Richardson. I'm a product developer with Enza Zaden, but I've got some history working in the breeding of buttercup squash. And I'm here today to talk about the genetic strategies we can be using in the management of soilborne disease for buttercup squash. So if I go to the starting slide, please. Before getting into the specifics on resistance, I would like to talk a bit more about what plant breeding actually is. So plant breeding is, there's a few definitions you can look for, but I think one of the most descriptive definitions of what it really is it's the art and the science of genetic improvement of plants for human benefit. And key to that definition is this idea of genetic improvement, which means you're actually looking for differences in the genotype of different individuals, and you're selecting these, and these changes are heritable over time, which means you have changes in the population of a certain trait over this time. And part of this are two fundamental sort of equations we look at, and I won't go too much into the equations, but it's quite important, is this idea of the breeder's equation, which looks at your response to selection, which is how much you're actually able to improve a trait over time. It's a function of your heritability, so how much of a trait comes from your genetic variation, and then also how much you're able to select from in your population, how extreme your selection can be. And that's a function of how diverse your population is. And when you're looking at selection and variation for any given trait, you've got your phenotype, which is what you can see or measure, is the product of your genotype. It's also a result of your environment and also interaction between these three. But typically within breeding, what we're trying to look at or select is the genotypic differences. And so when we do breeding activities, we need to be able to identify differences in genotype to make effective selections to change populations and the traits within a population over time. Next slide, please. And when you go approach a plant breeding program for any crop and for any set of traits, you've got to first identify your breeding objectives. You then need to, so this could be, say, for disease resistance, for example, but you then need to be able to find sources of diversity for this resistance. And this often means you're looking at a wide range of germplasms and doing screening to actually see if there is diversity and improvement for that trait of interest. You then need to be able to create genetic variation for this trait through combining this novel source of a trait with other sources to get new combinations of genetic material from which you can select. And then after that, you need a good evaluation process through which you can actually screen out differences for the trait you are looking to improve. And then you can select those genotypes, those individual plants or those families for that trait. And then you can do this one successively, your selection, your evaluation, until you have a new product, a new variety, a new cultivar at the end. That's got a unique mix of, say, the new traits you wanted to incorporate, plus all the other agronomic traits you require to be successful in a certain crop. Next slide, please. Now, to go to the point of how do you approach the problem of disease, particularly soilborne disease, there's two main approaches you can adopt through plant breeding. And the first is tolerance and the second is resistance. Now, tolerance is, these are often used interchangeably, and there's sometimes overlap in how they might be expressed or how you can see it performing in a crop, but there's some important distinctions to make. So tolerance is the ability of a plant to endure infection while still maintaining acceptable levels of growth and yield. So it's the ability of the crop to be in the field, it gets infected, it's still pathogen active, but you're able to get something out at the end of that crop period, that crop cycle. Versus resistance, where you actually have the innate ability of a crop plant to suppress the disease development and pathogen growth or pathogen growth. So the key difference here is your ability to manage pathogen loadings within a system. And so to sum us up, tolerance mitigates the negative impact of infection on host fitness without suppressing pathogen growth, whereas resistance actively limits pathogen proliferation. Next slide, please. So if you just fill out the, I haven't removed the animations for this one. Just go through them quickly. Thank you. So resistance in crops comes down to your ability to recognize pathogens, foreign invaders. And there's two main processes through which a plant or crop does this. Now, the first level is called pattern-triggered immunity, or PTI, through which a plant has a broad network of receptors which can recognize broadly conserved molecular sequences in a pathogen. So it might have receptors that can recognize chitinase, which is associated with fungal pathogens, or recognize flagellin, which is associated with bacterial pathogens. And this recognition then means it's able to cascade into an immune response, which is possibly able to stop the advance of the pathogen. Perhaps it can produce a thicker cell wall, which stops the aphosoria of a fungal pathogen proceed with its infection. The problem with PTI is that it can be circumvented by what's called pathogen effectors, which are specific molecules which can disrupt the recognition systems of PTI. And how, so this is, effectors can bypass PTI, but then you have effector-triggered immunity, where a host plant is able to generate a specific protein sequence or receptor molecule, which can identify a very specific effector associated with a certain pathogen. And this level of, and then this recognition allows it to, again, signal a cascade into starting a resistance response, possibly the hyperreactive response, hypersensitive response, sorry. And then that stops the pathogen development. But so to go over it again, you've got PTI and ETI. PTI works on a very broad level of pathogens, whereas ETI works on a specific race level. And when we talk about plant breeding and how we get resistance, whilst both mechanisms are relevant, ETI, the ability to have resistance genes or R genes, which recognize specific strains of a pathogen, is sort of like the gold standard for being able to resist pathogens. Next slide, please. Great. So the way that R genes work, it's often a gene for gene mechanism, which means you've got, say, a host plant and a pathogen, and then you've got a host plant might have an R gene, which directly relates to a virulence gene in a pathogen. The virulence gene in the pathogen will express an AVR protein, and then the AVR protein product will have a one-to-one, I guess, you got your R protein, which will match to an AVR protein. So in this example here, we've got the blue shapes and the red shapes, which are actually complementary. And this allows binding, which allows recognition in the host cell, which allows then a hyperreactive response to occur, and then you get resistance being induced. And an example of cucurbits is you've got your Fusarium oxysporum malonis, which gives you, you've got a FOM2 gene, which infers resistance against races 0 and 1 in melon, Cucumis mellow. Next slide, please. So we've also got qualitative as quantitative resistance when we're talking plant breeding. So qualitative is R genes. It's major resistance. It's your ability to have a very strong response to a specific race of pathogens. But it's also, we also have additive resistance, which is quantitative. And this is usually coming from multiple small genes with small individual effects, but they might have a larger sum effect. And this is generalized. So this is more aligned with the PTI system. If you could go to the next point, please. Thank you. So qualitative resistance is more effective usually, but the quantitative resistance is more durable. And we'll discuss that a bit more later. Now, when you're trying to breed for a qualitative or a major gene or an R gene approach through plant breeding, you really do need marker-assisted or molecular methods to do so. You can also do this. It is very helpful with a quantitative approach, but you can get by with just using bioassay or table test. Fundamentally, for both methods to work, you need to have a good table test, so bioassay development to actually screen genotypes for the resistance or the susceptibility to a disease. But that also means that that relies upon you to have good isolation, characterization, and verification of the causal pathogen. Now, this is, as an aside, I think it's quite interesting, is this idea that there's no, so you've got Buttercup squash, which is Cucurbita maxima. There's no squash cultivars on the market that claim resistance against soil-borne pathogens. You've got sort of some that do black rot, Didymella, but as far as the ones that we talk about, so the big ones saying Fusarium oxysporum, there's no claims there. And what that could relate to is you've got other species like watermelon and melon, which do claim resistance against Fusarium oxysporum, because there's been need to breed this resistance into these crops because they are innately more susceptible to these pathogens. And on the other side, Cucurbita maxima has historically been used as a rootstock in interspecific crosses with Machata because it's got an innate resistance ability within it. So compared to the pathogens that affect these other cucurbits. Next slide, please. And that goes to say is that there is quite a lot of potential within the Cucurbita maxima gene pool. When you're looking at where you get your sources of resistance from, there's several layers to it. So you've got your primary gene pool, which is your similar cultivars from the same species where you can actually get traits very easily and just through traditional crossing. Then you've got your secondary gene pool. This is looking at Cucurbita ecuadoriensis, where you can then get, say, still get genes through natural crossing, but you might have to, you got low viabilities, but you still get some viable dry seed out of that cross. And then on the third level, excuse me. On the third level, you've got your tertiary gene pool, which is your more distantly related relatives, but you can still get to a cross, but you rely upon embryo rescue and tissue culture at that point. And the main point of this slide here is to say that there is actually a lot of diversity available when it comes to breeding for, say, many traits. This is all mostly virus here, but for soil-borne disease too, for finding resistances to incorporate into a buttercup squash breeding program. Next slide, please. Now, R genes, so major resistance, it's very effective against specific pathogen strains, but in having such a strong resistance, you do create strong selection pressure for resistance breaking. And this accelerates the emergence of new pathogen variants that then overcome your R genes and make your resistance defunct. So if you can imagine, you've got like a mutation that occurs in your AVR protein, which makes the protein, that makes a gene, a virulence gene, virulent again in a pathogen. You no longer have recognition in your R gene, in your host plant. And that means that that single organism in that infected plant has a significant reproductive advantage over non-mutants. And then you get this incredible proliferation the next year of that gene within the pathogen population, because that is the only one that's been able to really reproduce from that previous season. Next slide, please. So then this poses the question of how do you actually increase resistance durability when you've got a, say, an R gene or a resistance gene in your breeding program. So there's gene stacking, just where you're incorporating several genes into one product. But then also you've got quantitative resistance, which it might not be so complete against a pathogen or a disease, but you do get more longevity out of that. Also, if you have multiple R genes, you can deploy them in different ways, like successively or in a diversified approach, or alternating different R genes, but ultimately pyramiding or stacking is the most robust to get more durability out of these. Next, please. And then also if you incorporate major resistance with minor or quantitative resistance, you also get more durability and longevity out of that resistance. But I think a thing to keep in mind is that whenever you have resistance, it's never going to be the be-all and end-all of your disease management program, and it has to really be part of your IPM holistic mindset, because it's all about reducing the pressure that's being put onto that resistance gene for mutation to occur. And that's where you have all your biological controls, your cultural practices, your chemical options too. Next slide, please. I won't go into the detail here, but basically there's a lot of steps to do a resistance breeding program. It takes a lot of time. And so starting with a new, even before you get to a gene you've identified, it's a big process to actually go from isolating your organism, identifying it, down to having a new variety released from which you can actually claim resistance against the causal organism. Next slide, please. So the second point is on tolerance. And with tolerance, it's, again, the ability to endure infection and maintain levels of growth and yield. So you can have indirect or direct approaches for this. An indirect approach is where you're looking at traits which are associated with tolerance. This allows you to do direct management of certain traits, but it's useful when direct measurement of tolerance is difficult. But also you've got direct selection. This is where you're screening genotypes that may demonstrate tolerance within the target environment. Next slide, please. So breeding for tolerance via indirect selection, you're usually looking at traits which can give you a bit more strength, a bit more power to maintain production, maintain growth, whilst infection is occurring. I think a good example of this is plant vigor. So thinking of wilt-causing organisms in many species of plants, it's often a numbers game, that the more you have the organism within the vascular system, the more it's going to take, the more wilt that's caused. But if you have a stronger vascular system that is, say, from more vigor plant, stronger vigor plant, then you're able to have a high pathogen loading within your host plant before wilt takes over, and then you have an impact on your fruit development. Keep going, please. Thank you. Other points too. So your canopy retention, your root vigor, earliness, these are all options you can use for your indirect selection for tolerance. You don't have to do selection in target environments, but the big problem is there may not be a very close relationship between your traits that you're trying to select for and the actual impact that these traits have on tolerance. So you don't always get the best advancement for your traits. Next slide, please. So when it comes to direct selection, this is screening genotypes in the field. Now, this is really an approach where you're looking for, you're not exactly quantifying the organism, but you're just putting it in the field and seeing what happens. Next point, please. So you need to target the trial environment, but a problem is that it's difficult sometimes to, we talked about it before, account for the genetic component of the variation you see. So because you've always got like a lot of variation in a field, and also in the pathogen loading in the soil, both in space but across time, always selecting on the genetic component of the variation can be difficult, and seeing that response to selection from year to year can also be tough. So to actually accommodate for this, you need to have really good trial design. So you need good replication, and you need probably sort of like quite large plots with a lot of variation still, maybe family level plots rather than individual plants you're doing selection on. Next point, please. And this requires you to invest in some really high resolution phenotyping approaches. So really, what I see as being a really powerful tool here is digital phenotyping methods. So you've got drone technology, which allows you to have phenotyping over a very large area. If we go through those points, please. You can have really high resolution on traits which are very hard to phenotype. So you've got it's high throughput, high resolution, but you can also look at traits such as plant height, canopy cover through time, but also looking at other parts of the spectrum. You can look at your chlorophyll activity, which you can really see the physiological function of your plant, even when it's not apparent if you're visually phenotyping. And the scalability of this really unlocks the ability to do good in-field phenotyping for tolerance trialing if you're trying to develop tolerant varieties for soil-borne disease. Yep. Yeah, so all those points there. Really, the power is that you get statistically powerful results from the trials, which are difficult to phenotype and difficult to sometimes get the genotypic variants from. Now, one point on tolerance, as I said before, is tolerance does not reduce pathogen pressure. So you do get, say, fruit yield at the end, but every season you're using a tolerant variety, you are still creating an environment for your pathogen to proliferate. And that means is that there's often a shelf life on the usage of tolerant varieties, especially in isolation. So again, tolerant varieties are more of I think, a short-term option for disease management through breeding, but also they need to be incorporated as part of IPM to really be viable and to get the most lifespan out of them. Next slide, please. Just a quick overview. This is like an outlook for different approaches for how you can do disease management through breeding. On the far right, we've got the most near-term options, which are probably the least effective as well as far as their efficacy. Like got late-stage screenings, early-stage screenings. Then you've got breeding for tolerance through your different methods, your direct and your indirect approaches. You've got quantitative resistance. And then at the very far end, you've got your qualitative resistance. And so you really want to be, I think, working with your options at each step of the process in terms of trying to maintain production through time. But the gold standard really is to have a good resistance package with, say, qualitative and quantitative resistance grouped into a good IPM strategy to have longevity and durable resistance and maintain production to the future. Next slide, please. Yeah, which is just the point there. You need to have your genetics are only as, you don't want your genetics to be the only point of your strategy, because if they're going to be overwhelmed, and then you're going to be back to square one. So it takes a long time to develop resistance. So again, you need all of your components of IPM to get the most out of a resistance or a tolerant variety. Next slide, please. So some final takeaways. We've got dual breeding strategies when we're looking at soil-borne disease, buttercup squash. You've got resistance and tolerance. There's a lot of genetic potential in the background and in the families, in the species around your buttercup squash to get resistance and tolerance into a variety. There hasn't been much activity of late because I think the challenges faced by growers internationally isn't necessarily the same as growers face in New Zealand. A key step is to actually have a good characterization of the pathogen. So isolation, characterization, and then developing a good bioassay to do resistance testing and screening with. We have our molecular tools, which are going to be essential for some aspects of breeding, especially for your more sophisticated resistances, but it's not necessarily always the case needed for more simple quantitative resistance. And tolerance breeding, it's a short-term solution. It requires less investment, but it's also a risk in the long term when it comes to your pathogen levels through time. And I think to really get good phenotyping is a big step towards getting good tolerant varieties, but also it can work towards your resistance breeding as well. And finally, again, to stress the point, it all comes down to having a really good holistic IPM approach, which you can slot your resistant varieties into to get the best out of new genetics. Anyway, that's all some references. And then next slide, acknowledgements. Just thank you, Aotearoa, again for having me to be part of the series, and also for Enza, Zaden, who've provided a lot of the context for me being able to discuss this point of soilborne disease and buttercup squash.
It's fantastic. Thanks, Hugh. I see there are a couple of questions in the Q&A for Hugh, and please continue to fire those through. If you preface the question with Hugh's name, and we'll endeavour to get Hugh to answer those and post them with the webinars when they're put up online. We've got a few questions now that have come through for John and
Thank you,
Mark Riley. So if I can just fire those through to you guys. Chris asks, do either of you test for beneficial microbes in relation to disease suppressive soils?
Well, we don't, so I'm not sure about John, but yeah, we're not offering that sort of test at the moment, but it's something we could look into, I guess. Also, I'd just like to clarify the answer I gave to the first question. The proportion of isolates that we identified morphologically was actually an incidence of each genera across all samples that we isolated from. So, for example, Aotearoa was on 90% of the samples, and, for instance, Phytophthora was only on 8%. So I hope that clarifies it a bit better.
Thanks, Mark. Question for John. How cost effective is the qPCR approach if you don't know what you are looking for in a random root symptom diagnosis?
That's where I think, and Mark touched on this, and I would certainly agree, is that, you know, for some things when we're testing, like we will test, for instance, beer and wine for a whole panel of species. And that's where those species are, you know, make up 80 or 90% of the most common problems that you're going to find. So I think if you have a very good idea of the species likely to be causing issues, where it might be half a dozen species, then qPCR can be a really useful tool. If you have no idea at all what it is, then it really comes down to that, as Mark said, a holistic approach or a combined approach of isolating that pathogen first, morphologically, or sequencing it, you know, which both Mark and ourselves do to isolate the species present, you know, and then saying, oh gosh, we do have a PCR for that, or no, we need to develop a new PCR for future testing. So qPCR is a lot faster than DNA sequencing, but often DNA sequencing, when you don't know what you're facing, makes the better sense there.
Thanks, John. Stephen asks, if it's a pathogen that has a root association, is it important to test the soil around the roots? He specialises in soils.
Well, I think it's good if you've got it out of the plant. then you've actually identified what's actually going on in the plant itself. Just because it's in the soil doesn't mean it's doing anything. So I think probably for me, it's more important to get something out of a plant which is symptomatic and obviously has signs of disease on it.
Yeah, sure. Thanks, Mark.
You may want to look at the soil in case you're wanting to replant or wanting to know a potential disease risk in future. But as to what's actually on that plant, yeah, I agree with what Mark said.
Yeah, the plant material. Yeah. Mark, do you plan to add genetic diagnostics like nanopore or metabarcoding to complement the current culturing process in the future? Would you see?
Sorry.
Answer that word first.
Yeah, well, we've thought about it and we've discussed it. I guess it's another form of sequencing, and it's something that, well, we could either do ourselves or get someone else to do it on our behalf. So yeah, we're thinking about it, but it's not there at this stage.
Okay. And the question goes on to ask, would you see value in using these as a complementary approach to capture the unculturable?
I guess that's true. Yeah, it would be useful in that scenario, I guess. Yeah, because I guess if you're sequencing everything that's there, then you might, as I said, you might pick up unexpected things, or as you say, things that are not able to be cultured. Definitely.
Great, thanks, Mark. Another one for Mark. During the pathogenicity testing stage, how do you make a call if a disease issue is caused by multiple pathogens, and how do you rank the primary and secondary pathogens?
Yeah, it's a very good question. Yeah, sometimes it's quite difficult actually, because if, like in that current, there were a whole range of things going on. I guess it comes down to experience really, and what symptoms you've got and what types of organisms you've isolated. So, and just making a call, I guess.
Great, thanks, guys. We'll move on to our next speaker, who is Dereck Ferguson. Dereck is an agronomy consultant in Hawke's Bay, formerly the agronomy manager for Brownrigg Agriculture. He has over 20 years experience managing horticultural and forage crop programs. Dereck is a member of the Vegetable Research and Innovation Board and holds several technical roles on research projects. He is the technical lead on the Aotearoa Soilborne Disease Management Project with the New Zealand Buttercup Squash Council, and is also a member of ALT's Industry Stakeholder Advisory Group. So Dereck's going to talk to us today about the Soilborne Disease Management and Buttercup Squash Project. Welcome, Dereck.
Thank you very much, Jeff, and good morning, everyone, and welcome to the Buttercup Squash Project. This is funded by the New Zealand Buttercup Squash Council and is done under the auspices of the A Lighter Touch program. Next slide. So, Soilborne disease in buttercup squash causes major issues in the industry. Firstly, lost yield potential, and also vine decline and canopy loss, causing sunburned and discoloured fruit prior to harvest, and also post-harvest decay in fruit. So that's decay that occurs in fruit that's been exported to the main countries it goes to, being South Korea and Japan. So that's decay occurring to fruit in those markets exported from New Zealand. So it's been calculated that the industry is losing up to 30% of production each year, and individual paddock losses can be much higher than that. You'll see a couple of examples in the right-hand photographs there of premature canopy collapse prior to harvest, and when fruit is exposed to sun quickly through canopy collapse, you get that white sunburning you're seeing there in the bottom right photograph. And as I said before, that's just one of the symptoms or the outcomes of Soilborne disease. The in-market decay can be also very severe. Next slide, please. So I'd like to talk to you about the project itself and the structure of the project. The central focus is the grower toolkit, and it has these 4 components around the outside which feed into that grower toolkit. So we'll discuss these sort of points one by one. There are, we conducted a plant breeders survey, and we're having dialogue with breeders like Hugh about soilborne disease and squash, and getting them to engage more on this, because it's become evident it's more of a New Zealand issue in Hawke's Bay and Gisborne than it is in some of the overseas markets they breed for. We've done grower surveys and around their attitudes to and adoption of biological products. And this is done for the grower toolkit and will be repeated. And growers have provided input for us into the things they want in that grower toolkit, what's important to them. This has been a 5-year project that ends at the completion of the current Lighter Touch program in March 2027. And really that grower toolkit, we're looking for that to be the enduring outcome from this project. Next slide, please. So that first component was the literature and knowledge review, and this was led by Lincoln University, in particular Dr. Helen Rees, who's sort of an expert in this soilborne disease field. And it had input from Ian Harvey from Plantwise and Lincoln, and Mark Braithwaite. We were fortunate to have Mark on the team there, because he could add his experience and knowledge, having done pathogen testing in Buttercup squash in Hawke's Bay and Gisborne there. And so he was able to discuss in that paper the probably 7 pathogens which he's worked with or discovered in squash in New Zealand. And this includes Fusarium, which is up to 8 species he's found within that in squash, and 3 of which are most common. Other sections in the review include comparisons with overseas squash production and the diagnostic tools that have been discussed today. Management options also include plant breeding opportunities, crop rotation, plant nutrition. So it was a significant piece of work. It's 125 pages, including its references and appendices, and it looked through both New Zealand and overseas literature, published and unpublished, and grower information and experience. Next, please. So trials were another component of the project, and there's a couple of facets to this. The first was we conducted some replicated small plot trials in the years mentioned in the slide there, and there were 2 sites for each year, one in Gisborne and one in Hawke's Bay. And we were investigating things like biological seed treatments, topical applications of biological products, and some synthetic products as well. They were applied either as a seed treatment or planting, or very early in the crop's life, intended to be single applications or interventions. We did see some canopy deterioration before harvest across these crops, but we didn't see any significant differences between the treatments regarding canopy collapse, root disease, evidence of that, marketable fruit, yield, or storage. And the hypothesis was that these diseases usually operate in a complex with primary and secondary pathogens, some stronger, some weaker pathogens, and there may be either levels that are too great for single applications of treatments, or the treatments can't cope with the complex nature of this, or there are other variables in the field overriding these treatments, and we'll talk more about that later. Next slide. So this has led us to looking at doing pot trials, which we are undertaking this year, where we're able to control more of the variables that we can't achieve in the field. So we've collected soil from a field that collapsed. It was actually one that was in John's table there. It was called Tuki Tuki, where it had very high levels of total Fusarium, Fusarium oxysporum, and Fusarium solani. So we knew from qPCR testing in the paddock that it had high levels. We knew from the crop that grew in that paddock it completely collapsed prior to harvest. And indeed, Mark also did some plant diagnostics on that and returned the pathogens that we are sort of expecting and high loadings of those high incidents. At this point, I would say that the qPCR testing we've done is really interesting in the soil with John, but it sort of hasn't been a linear relationship between high levels in the soil returning expressions of soilborne disease in the resulting squash crop. So, for example, we've tested ex-pasture paddocks that are going into a first-year crop of squash. They can return relatively high levels of total Fusarium, Fusarium oxysporum, and Fusarium solani, but the resulting crop is healthy, has strong yields and good market outturn. Compare that with paddocks that have a long-term cropping history, including a lot of squash, and they may have more moderate levels of total Fusarium, Oxysporum, and Solani, but the resulting crop would still collapse and may have in-market problems with it. So we think this is due to us not actually measuring the right strain or species of the pathogen we're going after, so we're not being specific enough. Also, other points that John touched on, the significance of the data when you get relative readings, what does it actually mean, or other factors that are actually critical to triggering the disease in the soil, so environmental or soil physical properties. So back to our pot trial, we're going to use the soil that's we know has caused problems previously. We're looking at some key biofungicides like trichodermas, bacillus, and Streptomyces liticus, that type of thing, ones that we've identified through trials and the lit review and conversations with registrants so far. So we're going to try those. Also, we're looking at cover crops prior to planting the squash in these pots. And you'll see in the central photograph there, that's actually the pots with the soil and growing a ryegrass cover crop over the winter period. So most of the pots are going to have that, as it's also a common grower practice before we plant the squash. Out of shot in that photo, we've actually got 15 pots that have got vetch in them. Vetch was identified in the literature review as being a service crop that is helpful in suppressing Fusarium in the soil and mitigating the presence of the disease and the resulting squash crop. So we've had a pilot study running this winter before the trial proper in spring, and the pilot was really to understand growing squash in pots in a greenhouse, the irrigation required, and how to do this. And we've certainly uncovered a few difficulties which we're overcoming now, prior to the start of the trial proper. Next slide, please. So another component of those 4 points outside the grower toolkit is the demonstration of best management practices. So this is really a proof of concept for us in a grower commercial field. So it's using a layered approach of inputs that have been identified in the literature review, related Elida Touch trials, including the pea and bean work that Suni Ching spoke about a couple of days ago. Also conversations with registrants and industry personnel. This includes inputs like biological fungicides, again, Trichoderma, Bacillus, Pseudomonas, Streptomyces, foliar fertilizers, biostimulants, humates, fulvic acid, silica, and calcium. So the alt demonstration was a sprayer width inside a commercial field. So effectively we had a grower control and our demonstration. There were 2 sites, one in Gisborne, one in Hawke's Bay. And product applications were done from the pre-planting, the soil incorporated, and then right through the crop's life. Most of them were additions to applications growers were making themselves, like as part of their fungicide program, and didn't require additional applications for the grower. Next slide, please. So here are some photos from the Hawke's Bay demonstration site from planting in the top left. Top right is about a month later. It would have been a very dry spring, and we had some difficulty germinating the crops. They required some irrigation, but then, as it does, Mother Nature turned around and gave us a lot of rain starting on Boxing Day. So we had a bit of surface ponding there, which also created some compaction in the field. We still got a reasonable crop, albeit with a slightly smaller canopy than normal there in the bottom left photo. But it gave us a crop with near industry normal levels of yield, and that's harvest going on in the bottom right there. Next slide, please. So in the Gisborne demonstration, we didn't get any differences between the grower practice and the Elida Touch demonstration, but there were some sort of mitigating factors there. First being the very dry spring gave split germination in the crop and split maturity, which often tends to mask canopy collapse because it happens gradually over a period of time rather than rapidly if the germination maturity was uniform. The grower was already using quite a lot of the products we were using in our demonstration, which is, I think, a good thing for that grower. They're progressive and using these biological products in particular, and biostimulants. There was one product in our suite that they didn't wish to use, and there were a couple of timing issues as well getting products on. So I guess the difference between the grower practice and the alt demonstration was narrower than in Hawke's Bay. I would note that the canopy still collapsed prior to harvest there, so there's still issues in the field there. Next slide, please. So in Hawke's Bay, we did actually get differences between our alt demonstration strip and the grower practice there. First thing you'd notice was a slight improvement in canopy longevity, which is important. So it stayed erect and upright, covering the squash for longer leading into harvest. And this resulted in less sunburn in the crop at harvest time. We took various measurements of that. The first column, if you look in the table there, is actually an on-ground assessment using quadrats of the number of sunburned fruit there. So you'll see in the demonstration, the third row down, that there was less fruit, at nearly 50% less that was affected by sunburn. The second column, you'll see that resulted in a higher yield, nearly 2 tonnes over sort of 15 tonne yield. So it's a decent numerical advantage. And the far column post-harvest, this is looking at some transects on the ground, counting the number of fruit left behind by the pickers, and they've left it behind primarily because of sunburn there, and you'll see a reduction in the quantity left behind in the demonstration there. There was no difference in the average fruit size, so the treatments we put in the alt site hadn't actually increased our average yield, if you will, but the advantage came through a decrease in fruit that was affected by sunburn. It wasn't statistical when you run it through a stats package, but it was very close to being statistical. Not entirely surprising given the variation we see in the paddock itself, both in soil type and expression of soilborne disease. It's patchy in nature, it's not uniform across the paddock. We're grateful to the suppliers who donated nearly all the inputs for the demonstration, which was considerable. We certainly learned a lot about setting up a biological program in terms of application, compatibility with products growers are currently using, including synthetic fungicides and other biological products. Next slide, please. Extension is a critical part of the overall project, and I'd certainly like to acknowledge Gina Jewell and the members of the Lighter Touch team for a lot of help in this area. It's not everybody's area of expertise. There's been a lot of output in terms of magazine articles, newsletters. We've done interaction with growers through surveys and the Vegetable Roadshow presentations to update on the happenings in the project. Next slide, please. So the development of the grower toolkit, the central piece of the project, if you will, this is currently underway, and there's quite a few contributors to this, including the likes of Bruce Searle and Kathy de Villiers from the Bioeconomy Science Institute, formerly PFR. So we do have some quite sort of good intellectual and grunt in this with a lot of experience. We're looking to incorporate the findings from the literature review, including crop rotations, use of service crops, biological controls and other products and cultural practices. Findings from our trial program, including the pot trial, the demonstration, and there's other associated work going on as well, like the qPCR soil testing, conversations with plant breeders and other experts. So the content's obviously critical, but the format is also important. It needs to be readable and usable for growers, and also referenced for further reading should they wish to dive deeper in a specific subject. There are good examples for us to follow as far as layout and outline in the industry already, like the sweet corn toolkit And there's also a frost control guide for vineyards that we particularly like the layout of. It's going to be updated before the end of the project, so it will appear in the coming months very soon, but we will update it because there'll be more findings from inside the project before the conclusion in March of 27. And as I said before, we're looking for this to be a sort of an enduring outcome or output from the project. Next slide, please. So in conclusion, but these are sort of the work-ons or work currently underway within the project. We've obviously got to complete that grower toolkit. Definitive pathogen identification. Mark has done a lot of work in the industry to now on identification of the soil-borne pathogens, but we need to be more specific, as he and John discussed, to be absolutely sure of which pathogens are pathogenic and which are not, particularly when you're talking about the fusariums there, because if you're going to identify it down to species level, but you need to know which strain beyond that to be sure of its pathogenicity there, which ones are primary and secondary, and which ones are the strong pathogens and the weaker ones. So that would also be a legacy outcome for the project, and it's invaluable because then if we know specifically which pathogens are doing the damage, people who have biological products can screen those products against those pathogens. People like John can use it for the soil testing, so we can increase the accuracy of that PCR test, and growers can use that as a risk management tool. And also for people like Hugh and plant breeding, if they know specifically which pathogen they're going after. Currently, the plant breeders find that very difficult because it's such a complex. We've got we're going to repeat the demonstration, and we're adding some layers to that because we're now this year going to use vetch as a service crop in the demonstration compared to ryegrass. We've got the addition of compost to improve soil biology and structure, and we're refining our inputs and timing. We have the pot trials, and we continue to have dialogue with people in the industry, including the plant breeders. The bottom 2 points there are sort of sidelines almost. They're not actually part of this project, but work that is going on in conjunction with ALT, specifically the minor use program for insecticide registration there. So I would say for me personally, and I think the project team, it's been a program of continuous learning and evolution. The more we understand, sometimes the more you realize you don't know. But for me, it's been enlightening about the disease triangle and the importance of our soil environment on the expression of these pathogens in squash. You may have that disease present in the soil, but you need other factors present. Often I think it's for squash crops, relatively long cropping history in these soils and poor structure through continuous cultivation, and resulting issues from that lead on to soil-borne disease. So it is the diseases are in a complex, and it is complex. Finally, I'd like to acknowledge the squash project team for all their valuable input, their lighter touch management team and team there, Gina, Jeff and co, and also the squash growers for their input, and the companies or businesses that have hosted our demonstration sites. Thank you.
Fantastic. Thanks, Dereck. I was just thinking back to 2020 when we were putting the plan together, and we got some comments like, wow, that's a big topic to take on. But I think not a good reason not to do it because it's a big topic. How are you finding grower engagement with the project? You mentioned some uptake already.
Yes, I think having surveyed some of the growers, it's a bit of a continuum because some are already using biological products and are keen to learn more about it and use more of them. Others perhaps have a more conventional approach, but are quite open to the biological products. So often they want to see sort of proof of concept that's actually working, and hence our demonstration there. I think, yeah, so the engagement's good, and I think I'm confident that with this grower toolkit, we'll be going back to them with some things they can actually implement and really look at as we understand this issue that would make a difference over time. not all of these things they'll be able to do. Like crop rotation can be a challenge here on the East Coast with a limited number of crops. So there are some things that are difficult, but at least they will understand the causal factors.
Yeah, great. And of course, it's a massive investment from the squash industry in this project, and it's great that they took advantage of the Light of Touch program to do that. So well done. I think we might have a question for you, Dereck, come through. A comment from Alan Kale. Very good presentations. Thank you all. And there's a comment here from Sony. We only found good relationship from qPCR tests to diseases in the field for second-year crops in a previous project we had completed. We have used qPCR-C as a tool to help select resistant materials, though. Okay, so just more of a comment there. I think we're well over time, but again, thank you to all our speakers over the series. We had Len Tesarero from Australia, session 1, Manjula Kilaranatna, who talked to us from Sri Lanka on nematodes, Suni Chung on Tuesday this week, and of course today, Mark, John, Hugh and Dereck. So thank you very much to all our speakers. Thanks to the behind the scenes team as well, Gina, Sydney and Craig. And just to reiterate again, all registrants will be emailed the link to the recordings when they're available online. If there's any questions that have come up in the meantime, or we haven't answered them, you can email them to us at news@lightofTouch.co.nz, and we will endeavour to answer those when we publish the webinar as well. Thank you very much.
Thank you.